Hello everyone, my name is Matthew. I'm an instructor here at Key Prep, and this is lesson 10 on cell reproduction. So what is cell reproduction? Cell reproduction happens in two different types of ways, which is mitosis and meiosis. Mitosis happens in somatic cells, which are non-sex cells, so that's almost every single cell in the body. And meiosis is similar to mitosis, but some of the steps are a little bit different, and it also happens in only sex cells. In this video, we will only be talking about mitosis. So in mitosis, there are four phases. This consists of prophase, metaphase, anaphase, and telophase. And before mitosis happens, interphase occurs, which prepares a cell for mitosis. And after mitosis, or towards the end of it, cytokinesis begins, which helps the cell finish dividing properly. So in mitosis begins at interphase. In interphase, the cell grows and replicates its DNA. It does this so when the cell splits into two daughter cells, there is the correct amount of DNA for both cells. Interphase is split up into G1, S, and G2 phases, which stand for growth 1, where the cell grows, S for synthesis, where the cell synthesizes and duplicates its DNA, and G2, which means growth 2, where the cell finishes growing to its full state before dividing. Prophase is the first step of mitosis, and the chromatids become visible as pairs, and the nuclear envelope and nucleolus both dissolve. In prophase, the chromatids become visible because the chromatin, which is also known as genetic material, condensed to the point where you can see the genetic material with the microscope. A mitotic spindle is also formed, which is used in later parts of mitosis, and this spindle is almost like a string like substance in the microscopic level. At this point, the DNA has already been replicated, so there is double the amount of DNA in this cell that there would be in a regular cell that's not in mitosis. The next step would be metaphase, and in metaphase, the chromatin or genetic material condenses even further into what is known as a chromosome, and these chromosomes become visible under a microscope. And the nucleus also disappears in this stage, and the chromosomes are released into the cytoplasm of the cell. The cytoplasm is like just the whole area of the cell. The chromosomes also align themselves in the middle of the cell along the mitotic spindle, and this phase is often used in medical research of cell development because this is the earliest stage that you can observe a chromosome and is often used to find chromosomal abnormalities. The next step would be anaphase, and in anaphase, the chromatids mentioned in prophase separate from each other and are also pulled towards opposite sides of the pole using the mitotic spindle fibers, also mentioned in prophase, and you can see this in the image provided. And these motions are driven by a motor protein, which helps the chromatids walk along the spindle fibers. And the cell becomes longer, and you will start being able to see the separation of the cells. The next step would be telophase. At telophase, the cell is almost done dividing, and the daughter cells start to establish themselves away from their parent cell, and they start developing their normal structures. The spindle fiber is broken down into building blocks, and there are two nuclei that form, one for each daughter cell. The nuclear membrane and nucleoli also start to reappear and form. The chromosomes begin to decondense and return to their looser, almost string-like form. Next is cytokinesis, and this is the division of the cytoplasm to finish forming the new cells. And this process may start as early as anaphase, but finishes shortly after telophase. Cytokinesis almost looks like the pinching of a coin in half using a rubber band, and this pinch action is also known as the cleavage furrow. So that's it for mitosis. Once again, this was Matthew, and I hope to see you again in another video. Thanks for watching.